Stem cells are?
Stem cells are cells that can divide asymmetrically to produce cells that go on to make tissues. So for skin, you need stem cells to make all the skin that grows over your lifetime. And they retain youth so that they can keep dividing over and over, and they don't become any particular certain cell type over time.
And stem cells can become more stem cells, or they can become any kind of cell.
And there's a few different types.
Right.
The two main classes are multipotent, which can make a few different types of tissues. These are cells that you typically get from an embryo, or you can go all the way back to age 0, and pluripotent stem, what are called cells, which can make any type of cell and any type of tissue.
And you can think of this sort of like as one of those trees, right? Like you have the pluripotent cell that can do anything, and then after a while, a little further down in the development, you have the multipotent cells, which are still able to transition into several different kinds of cells, but not all of the different kinds of cells.
Exactly. And so if you want to build a mini human brain or rebuild a kidney out of a skin cell, you have to wind the clock all the way back to 0 and start again. And that's called inducing a pluripotent stem cell state.
And that goes back to what we were talking about earlier, the Yamanaka factors. This is one of the ways that we've identified that we can turn a stem cell or any kind of cell from being a stem cell back into a state of pluripotency.
Yeah, let's just talk about Shinya Yamanaka because it's really quite an amazing story. So Yamanaka in Japan in the early 2010s, was in his lab was figuring out or trying to figure out how do you take an adult cell from an animal or a human and make it pluripotent. Because if you could do that, imagine you could build any tissue you wanted. And he was trying lots of different gene combinations and hit upon 5 genes that when put together worked to take, so I could take your skin cell now using Yamanaka's genes or Yamanaka factors and make a pluripotent stem cell line and rebuild you. I could even turn you, by the way, into a sperm and an egg and clone you, but I won't do that. So Yamanaka found the following genes. So KLF4, SOX2, CMYC, and LIN28. We use 3 of them, particularly in my lab. We'll talk about that later, OS and K for short. But this was a major breakthrough because finally you could take anybody's skin cell, which you could easily get from the cheek or a biopsy, grow that in a dish, reprogram them, which takes about a week once you put these genes in. And when they grow up into what we call a colony, which is an assemblage of about 100 cells, you pick those, and now you could grow basically anything you want from Matt LaPlante cells.
Well, and I've seen this in your lab where you've taken cells from a mouse, you've induced them into a state of pluripotency, and then you have trained them, for lack of a better word, to grow into brain cells. And now you have these little clusters of brain cells that look like little brains.
Well, we've done that for mice and we've done that for human. We have mini human brains, and we take these from old individuals, old normal individuals, and also patients that have susceptibility to Alzheimer's disease. And again, I think I want to talk about this later, but what we have the ability in my lab to drive aging forwards in those what are called cerebral organoids and even in reverse. And that's what's important really for being able to study the human brain without having to touch patients. And we can go a lot quicker that way.
Before we sort of go down that rabbit hole, can I take us back just a couple of steps to how this actually happens? Like, how do you take these factors and put them into a cell?
Well, it's actually really easy. Any high school student could do it. There are a number of ways. The old ways of doing it are you can pass electricity through the cells in the dish, and they take up DNA that encodes these Yamanaka genes. You can put them in what are called liposomes, so liposomes, which are fat, membrane-bound little balls. But the way we do it in the lab, which is the most efficient, is we use viruses, domesticated viruses that are typically used in patients for gene therapy. We use them in the lab. They're called adeno-associated viruses, or AAVs. We package inside those the Yamanaka genes and just throw them into the dish, and they infect all of the cells in the dish. And now those cells are transgenic. They carry these genes, and we can turn those genes on and off at will using drugs and see what happens.
And the implication here is that we can take these now pluripotent cells, and then we can use them to treat areas of the body where the cells just aren't acting the way that they're supposed to act anymore. But there's still a problem with pluripotent cells, right? Or induced pluripotent cells right now. And that is cancer.
Yeah, if you take the age of a cell back to 0 and allow it to just grow and put it into a mouse and probably a human, you'll get cancer. Teratoma is a particularly pernicious type of cancer, which can be, a hairy ball of mass, sometimes with teeth in it. You don't want that. So you have to be very careful not to put these pluripotent stem cells back into the body and just let them grow.
Which is why right now most stem cell therapies aren't using induced pluripotent stem cells. They're using harvested stem cells, and we can get those from a variety of places.
Right. Mainly what we're using, or what scientists and doctors are using, are what are called autologous stem cells. So cells that you take out of your body from various parts of the body, typically fat cells on your back or your hips, and then purifying them out, freezing them down, and or putting them back into the body in different places or into the bloodstream.
So we can just suck stem cells out of fat, out of adipose tissue. A lot of people think like, well, why would we even go through the process of trying to induce pluripotency if we can just take stem cells out of fat? But one of the things that I think needs to be said, and you've done some research on this, is that stem cells age also.
They do. They undergo epigenetic changes that make them less able to regenerate new tissues. And that's also true. You see that with gray hair and hair loss. That's the loss of the stem cells in the hair follicle. One other thing that would just happen recently is it was discovered that you don't just have stem cells getting old, they can even be kicked out of what's called the niche where they belong, they like to reside. And that's true for the hair cell. There's a fascinating video that I threw up on Twitter, and the stem cell, you could see, they captured it under the microscope, it got kicked out. Anyway, suffice to say, what is really going on is that these stem cells need to be slowed down in their aging process or even replaced, and we're only just learning how to do that.
The other thing that we can do is capture them at a much younger state. That's where cord blood comes in.
Right, and you can even do that for adults. The idea is that if you capture these cells when they're very young, or if you're in your 20s and 30s, you can bank them, put them in a freezer for later use, either to replace the blood if you have leukemia, or even using new technologies to modify them and grow new tissues, perhaps even replacing old blood when we get very old.
And a lot of people are doing this now. It's becoming more and more common for people to say, hey, look, I want to preserve my child's ability to use those stem cells later on, even if we're not exactly sure how they're going to be used, and so they're banking them. Your kids are in their teens and early adult years, but if you could go back in time, would you do this?
Yeah, we didn't do that because at the time it wasn't clear if they were useful, but now it's very clear that there are uses for them, especially if they get childhood cancer. And so I would do that. If I was a new parent, I would aim to bank those. How about you?
Yeah, I mean, so as you know, I haven't followed you down every longevity rabbit hole that you've gone down and that other people have gone down, but this is definitely 100% something that if my child had been born today, I would absolutely do. I would be banking their cord blood.
Yeah.
So if I did want to do that now, though, how would I go about doing that?
Well, there are companies you can contact, and often the hospital and the doctors you work with know how to do this, but you need to get ready. You at least need a few days, often a week, to get ready for this because you need to spin them down and put them in a freezer. But when you have a placenta, there are particular parts of the placenta. You can take the cord blood, you can take a thing called Wharton's jelly, which is this gelatinous substance made of hyaluronic acid where there's a particular concentration of stem cells you can also isolate.
We can also isolate stem cells from adults. Again, these are stem cells that have, gone down their trajectory from pluripotency often to multipotency. And so they're not, they're more aged, but sort of the earlier we do this, the better, right?
Yeah, these stem cells in adults don't just age and become dysfunctional. They can outcompete each other. They can grow faster. If you have mutations in genes like TET1, TET2, DNMTs, they grow faster and can actually outcompete the rest of the immune system. If you look at 100-year-olds, often they only have one or two instead of thousands of types of immune cells, which is a real problem if they get pneumonia. It's often why these elderly people don't survive an infection. So what you might want to do is to bank these HSCs, hematopoietic stem cells, when you're younger so that you can replace your immune system as you get older.
However we obtain the stem cells, researchers are using them for all kinds of diseases. They're being used to regenerate and repair diseased and damaged tissues in people with spinal cord injuries, type 1 diabetes, Parkinson's disease, ALS, Alzheimer's, heart disease, stroke, burns, cancer, arthritis. And the reason why I'm saying making this big list is because now, thanks to what I mean, like really, you've done this to me. Anytime I hear a big list of diseases and one singular thing that can address all of those diseases, I don't think of those diseases separately anymore. I think of aging. And there's a lot of implications here for the potential to use stem cells perhaps to treat aging. And a lot of people, like these other things we were talking about, are really excited about this potential avenue for anti-aging.
Well, they do have a lot of potential. First of all, we can make new organs using these induced pluripotent stem cells, or iPSCs, and even these autologous stem cells we're talking about that you can pull out from children and from adults. They can be used to replace stem cells that get old. But there's a real fundamental drawback to using stem cells to live longer, and that is that they often don't find their niche, their resting place. Even if you inject them into the eye, often they don't find their home. And in fact, one of the problems was when researchers did this to patients in Florida, there were 3 people who became blind when it wasn't done correctly. So there are real
that
problems.
coincided with an FDA order that said, hey, everybody, slow it down, be careful.
You can get some stem cell therapies now, but the majority of them have been banned for now. But what is really a problem is that if you can't get them to the right place, they're not going to rejuvenate the whole body. So what we're looking at now is how do you rejuvenate the existing stem cells and stop them getting old, or even reverse the aging of the stem cells in situ where they exist. And we'll get to that, but this is the reprogramming technology that should work not just on normal cells, but even stem cells to make them young, truly young again.
All right, and there's been a lot of research showing that stem cells can have a therapeutic effect in like specific parts of the body. But when we talk about this like aging umbrella in general, there was one study, actually 2 studies, a phase 1 study to phase 2 study from researchers at the University of Miami in 2017 that gave stem cell therapy, harvested stem cells from younger donors, gave them to older, frail patients, and small scale, but it showed improvements in the distances that these people could walk. It lowered the levels of cytokines. It improved their mental state, and they had a reported quality of life improvement as well.
Yeah, I mean, that's a remarkable couple of studies if it can be reproduced. But for now, what most people are using the mesenchymal stem cells for is in joints. When you wear out the joint and there's not a lot of collagen, injections of stem cells into those joints seems to be at least therapeutic for a short amount of time. Whether or not it's because the stem cells stay in there or they're secreting exosomes or other peptides, it's unclear.
So this is another area of research then that, again, probably people don't want to just walk into a clinic tomorrow and get a stem cell injection because, like you said, the stem cells might not even know where to go. But in the future, this is likely to be part of people's common anti-aging regimen.
Well, I'm sure. I think the future looks like this, that we can keep our bodies healthy by eating right, doing physical exercise, taking the right medicine slash supplements. And when that doesn't work and things fail, you can then rebuild the body, replace cells, put in new organs. And that way we'll live probably many decades longer than we can currently live.
And then there's one more thing that in the future we may be able to do. And this leads us to this thing that you're doing in your lab right now, and you had quite a few published studies on that are pretty darn exciting. We're talking about cellular reprogramming.
Well, this is the big one. We've been working for many years on slowing aging, but we've wondered, how do you get that to be reversed? Is there a reset switch in an old cell? And we think we found it. We're standing, of course, on the shoulders of Shinya Yamanaka, who showed you can reset the age of a cell back to 0, but that, of course, causes cancer. And if you do that in a mouse, it'll die within days. So that's not going to be a therapy anytime soon. I don't recommend it. But what a wonderful student, Wancheng Lu, did a few years ago in my lab was he found a set of genes that are Yamanaka factors that were able to reverse the age of cells and tissues in an animal and in human cells to a certain point, going back about 80% of age, but not to 0. So that this was a new, safe approach to reversing the age of the body without any negative side effects.
And he did this by, instead of using all 5 Yamanaka factors, he used 3 of them.
3. And the CMYC, which is the M, we definitely don't want that in there, or Lin28. These are what are called oncogenic genes, cancer-causing genes. So we left those out. And surprisingly, those other 3, O, S, and K for short, worked, not just in cells in the dish, but even in the whole animal. We focused on the eye because we thought reversing blindness would be pretty cool and could be a drug, a very quick drug to develop relative to the whole body. And it worked. Wan Cheng sent me a text of a picture of an eye where he also regenerated the optic nerve that was crushed. And then he went on to restore vision in mice that were given glaucoma, which is pressure in the eye, and even restore eyesight to blind old mice.
And let's slow this down a little bit and take people through the process, because we've gone from Yamanaka factors to restoring eyesight in mice. Let's talk about first the conditions that we're trying to be fixed were damaged optic nerve cells. And you used AAVs, these delivery viruses, to send these 3 Yamanaka factors into the cell. Once they were there, what started happening?
Well, to be clear, our genome has the Yamanaka genes in it, but they're normally only turned on when we're little embryos. So we had to reintroduce those 3 genes with a system where we could turn them on at will. And we were using an antibiotic to turn them on called doxycycline. We injected them with the virus into the eye. It's a very simple procedure. In fact, a lot of patients that are blind get this treatment already with other drugs. So it doesn't hurt, only takes 10 minutes. We put it into the eye of the mouse. We damaged the eye and asked, could those reprogrammed eyes that we reprogrammed for a month, could they regrow? And the answer was yes.
And not only did they regrow, but these mice that were effectively blind, you can do little tests and you can see that their eyes actually work like young mice eyes again.
Well, we put pressure in the eye to mimic glaucoma, which is a major cause of blindness in the world. Or we just let the mice age out to 1 year of age and they were blind, essentially. And we could tell that they were blind because we show them little images that move, lines actually that move from left to right. And if a mouse can see that, young mice will move their heads. It's called the optomotor response.
You can see videos of this. It's really cute. They're like watching a tennis match.
Yeah, we do that too, by the way. We also, of course, we measure the electrical activity of those nerves. We also measure the age of those nerve cells. We look at which genes are switched on and off. And the conclusion was that the nerves went back in time, back to the full reset to about 80%. Their genes, the gene expression patterns, as we call it, which is which genes are on and off, was reset to being young again. And the mice got their vision back, almost as good as they were when they were young.
So a lot of people will hear this and say, man, this is really cool. You can reset blindness in a mouse or reset vision in a mouse. Can we do this with other cells?
Yeah, it turns out that it's not just nerve cells that can be rejuvenated and sent back in time. We've done this for skin cells. We're doing it for muscle cells. We've done it for other parts of the eye. And in fact, the shape of the eye and the structure of the eye, it doesn't just go back in time, it actually rebuilds itself like it's young again. So we're super optimistic that this may be a universal mechanism of age resetting in the body that can not just be applied to different mouse tissues, but also in humans as well. And we're in the process, hopefully in a year and a half, of trying to restore vision in human patients that have lost their vision due to either glaucoma or genetic diseases.
So this is happening because it's accessing the backup copy, the original state of the genetic code that the cells are building themselves up by?
Well, the genetic code stays the same. The epigenetic code, which is the reader of the DNA, which we talked about in episode 1, that gets reset. So the structures of DNA folds that have gone awry during aging, this X differentiation process where cells lose their identity, that gets reset back to youthfulness. But how that happens, how do you get a loop of DNA to go back into a bundle and a bundle to go back into a loop? We have no idea how that works. We know that there's a backup copy of not the genome, but the epigenome, the structures that read the DNA, similar to polishing the scratches on a CD with the analogy we talked about in episode 1. But how does the cell do that? How does it reestablish that original software and reinstall the software in the cell? We're only just trying to figure that out now. Eventually, we will know how this works, and maybe we'll even develop a pill instead of a gene therapy that could reset the age, not just of the eye, but the whole body. And when that happens, certainly it's going to be a very different world. But until then, we'll work on vision, and then we'll see where we go from there.
Well, suffice it to say, there are several ways that we think that we can get the human body to do this, to reset the epigenetic clock. We can do it through cellular reprogramming. It looks like it's possible to do through exosomes. And you just mentioned pills. And it does appear that it's possible that this is what metformin is contributing to, and some of the other drugs and supplements that we talked about in an earlier episode. But I mentioned metformin because a lot of people are going to say, okay, all of this stuff is great. You've told me a bunch of stuff that probably won't be accessible to me anytime soon. There is a treatment regimen that is being tested out right now that starts with metformin, but a couple of other elements as well. Let's talk about that.
Yeah, this is my colleague Greg Fahey, Dr. Greg Fahey, and he's been working on anti-aging or longevity treatments for a while, and he hit upon a triple treatment. He uses growth hormone, which is known to raise blood sugar levels. We talked about that as a downside. But then he realized if he could include DHEA, a hormone that is depleted during aging, as well as metformin, he could mitigate the negative effects of growth hormone. And he gave that for 12 months to 10 healthy men that were aged 51 to 65. And lo and behold, when he looked at the biological clock of those men, they went back 2.5 years, which may not sound like a lot, but as I thought about it, if you could truly reverse your age every year by 1 year, that means you're not going to age.
All right, something that confuses me about this combinatory therapy regimen that Fahey used in this small-scale trial is that it takes advantage of human growth hormone, which earlier in this episode we said probably not a long-term anti-aging benefit. So what was the idea, do you think, behind using HGH?
Well, HGH had been shown previously to reconstitute the thymus. This is a little organ gland that's here in your chest that is important for the immune system. This is where your T cells, your immune cells come from. And growth hormone can stimulate the regeneration of this as it degrades over time. I'm age 50. I probably have what's called a lot of thymic degeneration and involution. It's a vestige of what it used to be. And growth hormone can stimulate that, but it comes with side effects, as we mentioned, not least of which is increase in insulin insensitivity or glucose intolerance. In other words, the cell doesn't take up the glucose from the body when you have a lot of growth hormone around. So he combined that with two diabetes therapies. One is metformin, which everybody knows. And Greg also showed actually in an experiment that he did on himself that DHEA, this hormone that some people take for longevity, can also control blood glucose levels. So that combination of three gave the best effect. But I should say that measuring the clock in the blood doesn't mean the whole body is being rejuvenated. That's just one indicator. So what I'd like to see Greg and his colleagues do is to measure aging of other tissues and measure it using different clocks, not just the Horvath or epigenetic clock.
We talked in an earlier episode about the proteomic clock.
There are a variety of clocks. There's glycan age, which are sugars that are attached to proteins that change over time. And there's one called immunoage. You measure various changes in the immune system. Altogether, these can be used to get a better picture of whether Greg's treatment is truly reversing aging or just some aspects of it.
But the really frustrating thing is, even with these clocks right now, we don't know what we don't know 5 years, 10 years, 20 years down the line. So it's a really good indicator if we can get these clocks to reset, if we can get these clocks to lower, like Greg did, at least in the short term. In the long term, do we have to wait for the long term?
Well, it depends. I know that Greg's research is ongoing, and that I was with him a couple of months ago, and he told me that repeated treatments over more than a year actually reduce age based on this blood clock, even more so, and people are going back by a decade, apparently. So super interesting time. I would say the jury's still out whether this is truly reversing aging or just some aspects of blood aging. But it's a very interesting time that we live in where people are ostensibly going back in time.
One of the commonalities, I think, in almost all of these things that we've talked about today is there are these early indications that something might have effects, and people are jumping over one another to get to them. And, some people are going overseas. If they can't get it here, they're finding doctors willing to do this. They're, finding suppliers from wherever on the internet they can. And it's understandable, right? Because people are suddenly awakened to this idea that aging may not be something that we have to tolerate in the long term. So they're eager, but there's still work that needs to be done.
There is, but what I think is really exciting is we wrote our book Lifespan in 2018-19, and we envisaged a future where people could turn their clock back and be rejuvenated. It's only been a couple of years since then, and there's been a huge advance in the ability to reprogram the body, to measure it, These tests are now available to the public to measure your biological age from blood and from spit. We're in an age where I would say it's hard to even predict where we'll be in 5, especially in 10 years from now.
But if you're frustrated, if you're really overeager, there are things, I think this needs to be said, like you can take steps right now. You don't have to necessarily be taking peptides and exosomes, and you don't have to, hunt down Greg and say, like, give me this combinatory therapy. Just slowing down the biological clock right now, doing the things that we talked about in the first few episodes is going to buy you that time for these therapies to mature to the place where we know that they're safe, we know what they're going to do.
Right. So stay young, stay alive, because the future looks bright. Just with the current pace of medicine and medical technologies, if you live an extra year, you get another 3 months of life. We could easily approach a time where every year you live, you get another year of life. And that's when the world becomes super interesting.
What are we talking about next episode?
We are talking about aging on the outside. So we've talked a lot about how to keep your insides young. Next episode, we're going to talk about your skin, your hair, your nails, how to keep those looking good. And it's not just superficial. It's really important. It's actually been shown that if you look younger on the outside, you're probably young on the inside too. So stay tuned for that.